Now we are recording. So, this will be the last chapter that we will cover. Okay, and honestly, it's just been so much fun there with you guys. I've had the core students there that have shown up every week, and I know that the rest of you guys are watching these videos and working and living your lives, and I'm just glad that I was a part of it. And what's going to go on there for next week, just to let you guys know there for the test, the test will not be open there for a week. All right, that's what I'm thinking there. The test will be open for about maybe 24 hours. That's all we can do. So if we have this test there on a Wednesday, if I open it up at 9 o'clock on a Wednesday, next Wednesday it will close in 24 hours. So next Thursday at 9 o'clock. So just checking the dates here. So next week's final quiz 6, okay, will open there December the 11th at 9 o'clock on Blackboard. No difference. The only thing is that it won't be open for a week. It'll be 24 hours. So you're going to have to motor. Okay. So do your best. Watch the videos. The PowerPoints there. All of the review questions that are located in the folder, they're there to assist you. So please have a look at them. The answers are there. Let me know if you guys need any issues, okay? Sorry, if you need anything, if there are any issues. If you guys need any issues. Who needs issues, right? What's going on? Am I missing anything there? Have I missed anything? Are there any questions there that you guys have for me? You raise your hand, let me know. Jesse, I saw that you raised your hand there a while ago, but I assuming that was for the you guys couldn't see the screen there before. But let me know, and let me know. Let's go, shall we? Now, oh boy, I'm going to share screen again. Let us save. Okay, so that is ready to go. Let's go. Please don't screw up. Come on. There we go. Okay, I hope there that you guys can see and hear me. Can I get a thumbs up? Oh, please let me get a thumbs up. Okay. Hello? Can you guys hear and see? Okay, thank you. And if you guys can't hear or see if there's anything there that seems out of miss, just take your microphone off and give me a shout. Okay. Where you guys are going, okay, after this course, I'm not even thinking this course. I'm not even thinking PSW. You guys go out and get your PSW and you get out there and work. Please come back and do a nursing degree. Georgian College is awesome. We've pumped out so many nurses and they're all out there and they're making the world better. Okay, PSWs are doing the same and we need more of them. You guys need a job reference. You guys need anything there from me. There's my email address. Shoot me an email. Get a hold of me. Just to let you know that if you email me from your Georgian College email, it will go to my inbox. I will see it. If you email me from any other domain name like Hotmail, Gmail, or I don't know, Phil's Fitness, whatever.com or whatever, it'll go to my junk mail. Okay, so I won't see it. So if you're looking for a job reference, email me. I'll let you know the procedure. And then if you do use my information there for a job reference, just give me a heads up when you anticipate them to call, because they're going to be emailing me from a different domain name. So their email is going to go to my junk mail and I won't see it. Okay, so that's what's going on here. My coffee is over on the other side of the room, so I'm going to go get it because I am useless without coffee. How sad is that, eh? Yeah. So I was thinking there that this PowerPoint was a little shorter, but it's 44 slides, so this is going to be an hour. So this is a good one here. So, and I don't really want to get too heavy into this, but we are going to sink our teeth in a little bit. Okay, there's our heredity, there's our genetics, which you guys have already had a little bit. Haploid, diploid, gamete, spermatogenesis. You guys are kind of already there. We're going to continue on with a little bit of chromosomal stuff. And then we'll look very, very mildly at some human development. Okay? I'm not an embryologist by trade by any means, but we're going to have some fun. So, to no further ado, I can see there that there's no hands and you guys can hear and see me, so let's go, shall we? Fertilization, sperm and egg unite. When? Near the mid-cycle, just after ovulation, right? Where? About the first third of the fallopian tubes. Remember the ampulla? That midsection there? That's where you want, okay, that's where you want fertilization to occur. How? At intercourse, the sperm swim to the fallopian tubes. At fertilization, the nuclei of the sperm and egg unite. Remember, 23 chromosomes for the egg, 23 chromosomes for the sperm come together to form the zygote, which has diploid 46 chromosomes as opposed to haploid 23 chromosomes. Okay? Lots of stuff there, but I think you got it. You got it. Prenatal development. There's 3 periods there. There's what we call the early embryonic, which encompasses there the first 2 weeks. There's the embryonic, which is weeks 3 to 8. And then you have fetal, okay, which is about week 9, just after 2 months or so, all the way up into birth. Okay, so there's different stages there that we'll have a look at. Pregnancy, this is just a word there that we use that encompasses there the entire 9 months, 38 weeks. Conception until birth, divided into trimesters. First 3 months, second 3 months, third 3 months. Prenatal development includes what we call cleavage, cell division by mitosis, growth, size of the cells increase, morphogenesis, shaping of the cell clusters, and differentiation, cellular specialization. And you'll see here that when we're going there, there's move in this direction right here. We've got A, B, and C, and D. Okay, so we're kind of moving in this direction here. There's a lot going on in this slide here. So you have zygote, okay, and there is the egg and the sperm. And we'll just move this up here, because there's lots of good stuff in here as well. All right, good. So there is your zygote formation, the fertilized ovum. Okay, the sperm have entered in. What was that on the head of this sperm head that allowed it to get into, was that called the acrosome? You remember that one? Good, good, good. So you'll see what's going on here is there's your zygote, and then you get what's called a blastomere. Okay, this is multiple cells there where you start to getting mitosis divisions, okay, rapid growth. You go from 1 to 2 to 4 to 8 to 16, okay, and then once you're at 16 cells, okay, it's not really called a blastomere anymore, it's called a morula, okay, call it a raspberry because it kind of looks like a raspberry. I don't really see it, but yeah, sure. And then you get into what's called a blastocyst, early and late, okay? And just reading in down here, during the passage through the fallopian tubes and through mitotic division, a zygote becomes a series of blastomeres. Entering the uterus there, the cluster of developing cells is called a morula, okay? The transformation from zygote to morula takes about 3 days, okay? Zygote to morula takes about 3 days. The trophoblastic cells secrete the what we call a human chorionic gonadotropin, which we've spoken about already, maintaining the corpus luteum, okay, and assisting there with implantation. All right. Yeah, that's pretty much it. Within the blastocyst, the cluster of cells. I'm not interested in inner mass cells, but there's more about your morula there. 16-cell cluster that resembles a raspberry. It floats in the uterus there for 3 to 4 days, becoming a blastocyst, and that's what we've got going on in here. Okay, twins. There's monozygotic. This one zygote. You see how that strange English works here. Okay, these are what we call identical twins. They develop from the same zygote. They have identical genetic information. One egg, one sperm, the zygote is created and then splits into 2 distinct human beings. Okay, dizygotic, fraternal. These are 2 individuals, like the mom released 2 eggs, and 100 million sperm came up into the fallopian tubes and implanted both of those. Okay, impregnated both of those. So therefore, mom is now carrying 2 different individuals. Okay, could be boy, could be girl, could be girl. Okay, different DNA, different people. Here, identical twins. 1 egg was produced, 1 sperm impregnated it, and then for some reason there, this zygote split into 2 identical individuals. And we were talking about that there earlier in the previous PowerPoint. The whole idea that I read in the paper there that these 2 identical twin sisters, one of them needed a kidney. Normally, when you receive an organ transplant, you have to take immunosuppressing drugs, okay, to suppress your own immune system for the rest of your life, because your body will begin to attack that foreign organ. But because that came from your identical twin, you're good to go. All right? Lucky, lucky, lucky. So how twins are made? There's your fraternal, okay, dizygotic. 2 sperm fertilize 2 separate eggs, which develops in the womb at the same time. Okay, DNA is 50% maternal from mom, 50% paternal from dad. So 2 different eggs, 2 sperm, 2 different individuals, different DNA. Identical twins, monozygotic, okay, where a single sperm fertilizes a single egg, which then divides and makes 2 babies. DNA, 100% maternal, 100% paternal. Interesting. Semi-identical twins, where 2 sperm fertilize 1 egg, which divides and makes 2 babies. Strange, eh? So in this case here, the DNA is 100% maternal and only 78% paternal. The semi-identical twins in the first known case had 50% paternal DNA. Interesting. So in this case here, 2 sperm can impregnate 1 egg. Embryonic period there, extra embryonic membranes. And you'll see what's going on here. Now we have development there of the baby. Okay, so now we have 2 organisms in one body. Okay, we have what's called the amnion, amniotic sac, okay, and amniotic fluid. You'll see it right in there. There's your amnion and your amniotic sac. And inside, your amniotic fluid. Okay, chorionic villi, and your placenta. You'll see there's your chorion on the outside. Okay, placenta right in here. Yolk sac, this is for red blood cells, immature sex cells. Okay, and you'll just have a read here. The developing baby floats in amniotic fluid formed by the amnion. The baby's urine also helps create much of the amniotic fluid, about 1 liter or so of that amniotic fluid. Okay? This amniotic fluid protects the embryo, which also drinks and digests the fluid. The embryo secretes waste in the cells in the amniotic fluid. This is the basics for the diagnostic test, amniocentesis. Rupture of the bag of water often signals the onset of labor. So when this amniotic fluid starts to leak out, mama knows baby's coming. Okay, so at birth the membranes are expelled with the placenta as after birth. Okay, and yeah, just the same slide. I just took that information and put it up in there. So great read there. You have a look there. There's your chorion, and then there's your amnion, amniotic fluid. There's your embryo. There's the yolk sac. Okay, lots going on, and then there's your umbilical cord right in there that connects baby's belly button, as we all have one, to mom. These diagnosis tests there for genetic defects can be done by sampling the amniotic fluid or the blood in the chorionic villi. In both tests, embryonic cells can be examined and extracted. So you'll know there that baby drinks, pees into this, and we can detect a lot of information from an amniocentesis. Okay, and you'll see there you take some of the amniotic fluid, take it out, and give it a test, and away you go. Okay. Chorionic villi and placenta. Okay. The placenta formed when the chorionic villi penetrate the endometrium. Okay. That's what's going on there. When the egg comes down and implants itself, that is the site there for the umbilical cord. Okay. Site for fetal eating, breathing, excreting, secretes hormones. Umbilical cord joins the fetus and the placenta at this site. Okay. The finger-like chorionic villi penetrate the endometrium. They sit in the blood-filled spaces called lacunae. You'll see them in here. Okay. The placental membranes ensure that the maternal and embryonic blood supplies are separate. Now, does that mean there that there's no movement across? Oh, there can be. Okay. But generally speaking, the mom's blood and the baby's blood are kept separate. In theory. The umbilical cord connects the embryo to the placenta. The cord's blood vessels carry unoxygenated blood from the embryo to the placenta, where it is oxygenated and returned to the embryo. So you see the baby's blood doesn't make it all the way to the mom's lungs. The baby's blood makes it to here, where it is oxygenated and then taken back to baby. The two roles there of the placenta: the site across which nutrients and waste are exchanged between baby and mom. Acts as a gland. It secretes hormones that help maintain the pregnancy, prepare the body for birthing process, and promote postnatal events such as breastfeeding. Okay? And excellent. Good little movie here, embryonic period. Organogenesis. The formation of organs. Embryonic disc, formed by blastocyst in inner cell mass. Inner cell mass there forms 3 primary germ layers: ectoderm, mesoderm, and the endoderm. All right, I encourage you there to have a watch of the movie there, and that is all the organs and tissues of the body develop there from these 3 germ layers. Okay, just zooming in here, you can have a look. The ectoderm gives the nervous system portion of the special senses and the skin. The mesoderm gives rise there to muscle, bone, blood, and many cardiovascular structures. The endoderm gives rise to the lining there of the digestive tract, respiratory tract, and parts of the urinary tract. So these 3 here, they're off on bigger and better things. These are the starting layers there for basically all the organs and what goes on there later on in an adult person. They all start with these 3. Have a watch of the movie. It's a good one. Oh, there's my cat trying to get in. Oh, cats. Embryonic period there. Teratogens. Oh boy, these are events, drugs, things there that can interfere, okay, with normal prenatal development. German measles, IV drug use, alcohol, that one there, we do know that drinking while pregnant will cause devastating effects there to baby. Radiation as well. There was a drug there that was introduced in the 1950s. It was called thalidomide, and it was a drug there that was synthesized there to stop or reduce morning sickness, okay, in newly expectant moms, okay, and you can see there that this drug taken at certain times there interfered there with appendage development, okay, and you can see there the babies were born there with no arms, no legs. They were born there with hands attached to their shoulders, feet attached to their hips. Yeah, quite devastating. Okay. Fetal period there, when we're talking development there, the body proportion changes. You'll see there that babies tend to be quite strange, and I don't know. I don't mean that in in any type of bad way. But if you look at a normal adult human being, an adult's head is about 1/7 or 1/8 of their entire length. Okay, babies' heads though are about 1/3 of their entire length. So the head has been overly developed there, so that baby is ready to go there by the time it's born, and the head there is about 1/3 of its entire body length. The fetus is viable at around 20 weeks. Meaning there that baby can actually, baby is ready to go in some sense. Preterm birth there to before 38 weeks. Okay. Just some words here that you might see on a test there next week. Quickening, movement of fetus felt by the mother. So when baby kicks his feet, that's what they call quickening. Lanugo. Fine, very fine hair that covers the fetus. It comes off there after baby's born. Vernex caseosa, white cheese-like substance protecting the fetal skin there from the amniotic fluid. So baby will be encased in this, okay? It protects baby's skin there from the amniotic fluid because he's been sitting in the amniotic fluid there for 9 months. Abortion. Loss of the fetus there at any time, can be spontaneous, which we call a miscarriage, can be induced, can be therapeutic as well. All right. Maternal changes in pregnancy, the rate of metabolism there absolutely increases. Mom is now eating for 2, carrying 2, walking for 2, sleeping for 2. So mom's rate of metabolism there absolutely increases. Blood volume expands to 40 to 50%. So now mom is carrying, well, 50% more blood there than she normally did. And how much blood do we have? How many liters? About 5 liters. So now mom has about 7 liters of blood running around. Kidneys produce more urine, of course. Nutritional needs increase, pickles and ice cream, which is absolutely true, by the way. Trust me, I know. Increased hormonal secretions there. There's your human chorionic gonadotropin, estrogen, progesterone, prolactin, oxytocin, prostaglandins, thyroid hormones, aldosterone there. I like all of these ones in here. Okay, love them. And there's 2 thyroid hormones there that you should know there for the bones, calcitonin and parathyroid, the control of calcium. Labor, birth of baby. So forceful contractions, they expel the fetus there from the uterus, and that all begins there by baby's head pressing, because baby is inverted, right? Head is below, the bum is above. So when baby's head starts to press on the cervix makes a phone call upstairs to the hypothalamus. The hypothalamus there makes a phone call to the posterior pituitary, which then secretes oxytocin. And this one here gets those myometrium, okay, the muscle there of the uterus, to start contracting. Okay. Postnatal adjustments in baby. Breathing is established. Okay, excuse me. Because baby hasn't ever taken a breath before. Baby comes out, takes a breath. Breathing is now established. Got to get that oxygen in there, right? Blood flow through the lungs begin. Mechanism for temperature regulation adjusted. Functioning digestive system produces mechanonium. Okay, baby's first stool, which tends to be weird and green. Okay, because they haven't really eaten anything. Okay, most organs begin to function more efficiently. And there's some good reading down in here. You guys can have a look at that. Developmental stages, lifespan. Okay, you got the neonatal period there, birth to 4 weeks, infancy there, 2nd to 12th month. Okay, childhood, and again, here we're talking post delivery, right? Baby has been born. So the neonatal month, okay, birth to 4 weeks there, it's quite different there. You have to be very careful. If there's ever going to be complications, it's going to be exhibited there in this time period there. Okay, but it's the first month is the most precarious for any child. Okay, but if they can get out of the first month, then their odds of surviving are much greater. Okay, yeah, and you see here the infancy. It covers there the 2nd month to about the 12th month. Okay. Childhood encompasses there the 2nd year to about puberty. Adolescence there is puberty to adulthood. And adulthood there, whenever that starts. You ever see that commercial there? What was it? Adulting, right? What was that insurance company? Anyway, blah, blah. We'll talk about that there, probably never. Adulting. Whenever that starts, I still feel that I haven't hit that age yet. But adulthood there is adolescence there to old age. And then you've got senescence there, old age there to death. So that's kind of, I went into some prenatal development. I went into some postnatal development there. It's not a huge factor on this upcoming test, but there will be a couple questions on it. Nothing too crazy, okay? Moving on into DNA sequencing and hereditary, the whole idea of passing information on from parent to child. Okay, here's a beautiful shot there of a chromosome. This is our DNA. That's all just been nicely wound up. Kind of, it looks like a sweater. The yarn there from a sweater. And so the whole idea here, transmission of traits from parent to child. Color of hair, eyes, dimples, RH factor, earlobes, anything there that makes you had to have come from the parent. Okay. DNA, location of genetic information, chromosomes, thread-like structures of tightly wound DNA in the nuclei of most cells. Okay, so here's a shot there of your cell that we looked at there. Whoa, I think that was way back in week 2 or 3, right? And you remember there your nucleus, remember your nuclear pores there, though, and the whole idea that your DNA is inside. Okay, genes. These are found on the DNA, and you'll notice there these are genes. These are not GEAN. These are not the genes you wear. These are segments of the DNA that carry information for that particular trait, whether it's eye color, hair color, dimples, anything like that. It's all about the genes, and that's what's going on. So here's a stretch of your DNA, okay? And here, this stretch of DNA codes for eye color. This stretch of DNA here codes for hair color. This stretch of DNA codes for skin color. And these are what we call genes. Genes. Okay? So, getting onto genetics there a little bit here. What is genetics? The inherited traits which are determined by DNA sequences that are comprised of genes. Okay? The field of genetics investigates how genes confer specific characteristics that affect health or contribute to our natural variation, and how genes are passed from one generation to the next. Okay? Mutations can occur when the gene's DNA sequence changes. Now, you look at those teratogens there that we were looking at there earlier. Smoking, radiation, working over barbecues, all these different things there can cause the gene sequence to change. And just going back in here for these genes, that is a specific sequence. You mess with that sequence, you're not going to get what you're looking for. So the sequence of the DNA is absolutely imperative to maintain, because any type of mutations, alterations, deletions, additions to this will screw everything up. Okay? Mutations occur when the gene's DNA sequence changes. Environment can influence how most genes are expressed. Okay? Now, the whole idea there is we are who we are. Is that because of our parents or the environment? And you've heard about this, nature versus nurture. So the whole are we who we are because of our DNA, or are we who we are because of our environment that we were raised in? Like if you were raised in, I don't know, the north, the south, dry climate, wet climate, you were, the whole idea. Does that really affect who we are? And in some cases, it really does. Okay. There are thought to be about 70,000 genes in the human genome. That's it. Sexually reproducing organisms make up two types of cells. Gamete cells, we've already know about. These are the sex cells, right? Sperm and egg. They contain half of the chromosomes. They are haploid, okay, which we signify as a single N. Diploid would be 2N, okay? Diploid cells would be 2N, okay? 46 chromosomes, 23 chromosomes. Sperm cells and ova are gametes. Now, here we're talking meiosis, right? This is the whole process there to ensure genetic variability. We do not want these sex cells having the same sequence, because if there's a disease out there that attacks one of them, it will attack both of them. So we don't want any individuals who are exactly the same. Somatic cells, these are not your sex cells, sperm and egg. Somatic means body. These are your body cells, bone, muscle, skin, all these kinds of things here. Here we're talking meiosis when we're talking growth and development. Mitosis when you're talking growth and development. Gametes, meiosis. Somatic cells, mitosis. Okay? Where the parent cell divides into two identical cells. That's how the body grows. Gamete cells, these ones here, they develop through meiosis. The end result is different gametes. Somatic cells, the end result, exactly the same. We just want more of them. Okay? Diploid number, 2N, skin, brain, bones, muscle. Good. So gametes there, the male gamete is the sperm and is produced in the male gonads. There, the testes there. Okay, you remember those different kinds of cells. Okay. The female gamete is the ovum. Ova is plural and is produced there by the female gonads in the ovaries. Fertilization. During ovulation, the ovum is released from the ovary and transported to the fallopian tubes. We've covered all this there, where fertilization occurs there in the ampulla section of the fallopian tubes. Fertilization results in the formation of the zygote, the fertilized egg. Chromosomes exist in pairs. 23 pairs, one chromosome from each male and one female. So 23 pairs of chromosomes gives us 46 total. 23 came from mom, 23 came from dad. Okay, so you have 23 pairs. There are autosomes and one sex chromosome, one pair. So you have 22 pairs out of your 23 that you have. Now remember, we are 2N, we are diploid. We have 23 pairs of chromosomes, 46 total chromosomes. That's what makes us us. Of those 23 pairs of chromosomes, 22 of them are what we call autosomes. These are non-sex chromosomes. Okay? Don't have anything there to do with sex. The only one, there is one, and that's called the sex chromosome, and there is one pair, okay, that will either be XX, indicating female, or XY, indicating male, okay? So this is the whole idea here. There is 23 chromosomes in here, 23 chromosomes in here, and 23 pairs of chromosomes. You get that? 23 chromosomes gives us 23 pairs of chromosomes, which is 46. Okay? So, the human zygote is diploid. 2N is 46 chromosomes, 23 pairs. Okay? Meaning it has two matching homologs per set. Okay? Homologs, pairs of DNA copies. So it has 23 homologs. Okay, each homolog, N is 23, comes from each parent. Most organisms are diploid. Humans have 23 sets of chromosomes, meaning humans have 46 total chromosomes. Normal number of chromosomes is referred to as euploid. Aneuploidy, though, is the presence of an abnormal number of chromosomes in a cell. So we should have 23 pairs of chromosomes, 46 total. That's all right. For example, though, a human cell having 45 or 47 chromosomes, it does happen instead of the usual 46. Here, this individual has Down syndrome, okay, and which means there they have an extra 21st chromosome, all right. We'll look at those there more at the end. Chromosomes and genes are paired. They're the chromosome chart. And there is a chromosome chart where they look at every single pair of chromosomes and they can take pictures of it and they put it on display, and we'll show you that there. It's called a karyotype. All right? It displays the 23 pairs of chromosomes in a human somatic cell. Human somatic cell. That should make sense. Okay, so they take one of your skin cells, they take a sample, and they look at the chromosomes inside of it there. You'll have 23 chromosomal pairs. They take pictures of them, and we'll show you that there in a second. They're out of those 23 pairs, you have the 1 to 22. These are what we call the autosomes. They carry genes for most traits, but do not carry the genes that determine sex. Okay, eye color, hair color, all this is all carried there by these autosomes. The sex chromosome, called the X and Y chromosome, and you'll see there that the Y chromosome doesn't look like a Y. Okay, it determines sex. The female has double X, male has X and Y. Now, and you look at this there, I'm just going to zoom in on this X and X. Now, if, like for my daughter here, one of these X's came from me, and one of these X's came from my wife. So you'll see here, and in this combination, X and an X, okay, now we have a female. In this case here, you have this X came from my wife, the Y came from me, and you'll see there that in this scenario here, we have okay, my son. Now, there are other factors associated with that chromosome. There are other factors associated with that chromosome. There are diseases. There are troubles there within the DNA for each of these. Now, if you look at this individual there, you know, it's we have the same chromosome in different ratios and proportions, but you'll see there that this one here came from mom, this one here came from dad. If there is a disease on this chromosome, well, if there's some trouble with this chromosome, well, I have the exact same chromosome here that's in good shape. So this good shaped chromosome kind of masks and covers up any trouble there that's in this chromosome. Okay? So this is where females tend to have the advantage over males, because if one of those chromosomes is whacked or there's trouble with it or something's going on with it, there's another of the same chromosome there to cover it up. Okay? Now, so mom may have a disease, but you may not see it, because it's being protected by this other chromosome. This other chromosome, which is non-disease, is covering up this diseased chromosome. Okay? So that's where females have the advantage over males, because in this case here, if there's an issue on that chromosome, which came from mom, which was the same one here, if there's an issue with that chromosome, males don't have another X to cover it up. They have a completely different chromosome. So that's why some of these diseases there can be exhibited in males, okay? Because males don't have that extra X to cover it up and mask any trouble that's in this one. We'll talk more about that there later. All chromosomes except Y carry hundreds of genes. Somatic cells contain 2 copies of each autosome and 2 copies of each gene. Gene copies on 2 like chromosomes may be identical or different. And we'll show you this in more detail here. You'll see here that here's a picture of the X chromosome. Okay, double X there indicating female, and there's your X and your Y chromosome indicating male. Now, and when you're talking about female-related diseases there, we're just talking about sex-related, where if there is any trouble on the one X chromosome, females there, they have another X there that will protect and cover up any issues that might be involved with this chromosome. It happens all the time, okay? But even though the female may not be exhibiting any diseases, this chromosome here still has trouble. And when mama there is, making eggs there to make babies, if she uses this chromosome, baby will not have any trouble. But if she passes this chromosome off to baby, then baby potentially could have trouble. Okay. For males here, okay, there's your X and then there's your Y. This Y chromosome has none of this information in here. So any mal-information, any disease information in here cannot be covered up, and it is exhibited in males. Okay. Moving on. So, displaying the paired chromosomes, this is that karyotype, what I was talking about. And what they do is they go in and they just take pictures. Now, these chromosomes aren't just lying side by side. What they do is they take pictures, they cut and paste them, and they put them side by side for a nice, beautiful little shot here. So you can actually see if there's any type of issues going on with these chromosomes. Okay. Another shot there. Each homologous set is made up of 2 homologs. Now remember, if this is somebody's DNA karyotype. So there's 23 chromosomes. What are they? 23. So 1 to 22, these are your autosomes. Okay? These don't have anything to do with sexual characteristics, okay, and sex differentiation. These just have to do with physical phenotypes, stereotypes, sorry, not phenotypes there when we're talking about hair color, eye color, dimples, these kinds of things. This one here, chromosome 23, that is your sex chromosome, okay? And in this case here, you're looking at a female. Here's a shot there of a female as opposed to a male. There's your X and there's your Y. Okay. Meiosis is male. Meiosis in males is called spermatogenesis. Meiosis in females is called oogenesis. Produces sperm and ova. And we've seen these slides there before. I don't want to go too heavy into them there, but there's your primary spermicide. And you get the first meiotic division there. And you'll see there 46 chromosomes splits into 23 and 23. And then these guys here double. And this is where the crossing over happens. Okay, and I implore you guys there to look at the video there that went on in the last PowerPoint. There's usually a question or two on crossing over on the test that's coming up. Okay, and it's this crossing over that makes these 4 sperm completely different and never to have been seen in the known universe before. There is meiosis going on in oogenesis there, egg production, primary oocyte divides. Okay, there's your first meiotic division, and the end result there is a secondary oocyte and a polar body. And these polar bodies, as you know, just get the bin. So in this case here, the objective of oogenesis is not to make 4 egg cells from a primary oocyte. It's there to make 1. Okay. Now, talking about genes. Now, if this was like at the conclusion of meiosis, each haploid gamete has one of the 4 chromosomes shown. You remember there when we were talking about these sperm, right? And how they were all different from each other. You'll see what goes on here. One of those sperm will have this gamete. One of the sperm there will have this. Okay, it's different, right? You've got different things coded. This is the DNA, and you've got different things going on here. And then this one here is different, and this one here is different. So at the conclusion of meiosis, each haploid gamete has one of the 4 chromosomes shown. 2 of the chromosomes are recombinant. They carry new combination of genes. And you'll see there, paternal, okay, it comes from, and then you have maternal. All right, and it's just this whole idea of these genes crossing over, okay? And when you're making these right here, these guys here split, and then the DNA from these 2 here, there's this crossing over that happens, so that these sperm cells here ensure that they are all different. And you'll see there 1, 2, 3, 4, sperm 1, 2, 3, 4. They all look different, and that's what you want. Okay, and you can see what these alleles code for. Big H, brown hair. Little h, blonde hair. So this individual here will have blonde hair. These 2 individuals here will have blonde hair. These 2 individuals will have brown hair. And then you've got big E for brown eyes, small e for blue eyes. And you'll see there that each one has different combination. So these 4 individuals here are quite different. Modes of inheritance. Patterns of gene transmission in families are called modes of inheritance. These can be used to predict probabilities of traits occurring in offspring. There is dominant and recessive inheritance. Do you remember there? We're looking at big H and small h, big E and small e. Generally speaking, the big, that is dominant. Small is recessive. And how this works is, it's much better if you're in class there, but the dominant and recessive inheritance there, a dominant allele So here we're talking about a stretch of DNA that codes for a particular trait, whether it's blue eyes or brown eyes. But you'll see there that a dominant allele will mask the expression of a recessive allele. So you'll see here, a dominant allele, and I'll call that big B, and a recessive allele will be a small b. And let's just say there that the big B, this one here codes for brown eyes. Now, what goes on here is for any type of what we call phenotype, blue eyes, brown eyes, whatever you can see, one of these genes came from mom, one of these genes came from dad. Okay? And you'll see here in this case here, this individual will have brown eyes. Okay? Because that's all it's got. It's got brown and brown. Okay? And brown is a dominant allele. Look at this individual here. You have a brown and a blue. So this one here came from mom, this one here came from dad. This individual here has both genes, but because brown is dominant, this individual has blue eyes. Sorry, this one here has brown eyes. Oh my gosh, I need coffee big time. Again, dominant alleles, they mask, they cover up the recessive alleles. So in this case here, this person has a brown gene and a blue gene, so this person here would have brown eyes. All right? Because the dominant covers up the recessive. Now, when you're talking recessive genes, okay, the only way to get blue eyes is if you need both of the genes. Because we saw what happens there, a blue eye and a brown-eyed gene. The brown-eyed gene is what you see. It covers up the blue. Now, but if you have both blue, okay, this is a recessive gene, it only comes to face if you have both of them. Okay, so one came from mom, one came from dad. There's your blue eyes. So an allele that causes a trait or disease can be recessive or dominant and inherited in either an autosomal or linked sex manner. Autosomal conditions, remember that 1 to 22, affect both sexes. X-linked traits mainly affect males. Okay, remember what we talked about that, right? The whole idea there that if, I'm just trying to find that slide here. If there's a disease on one of these guys here, females have another one exactly the same that will cover it up. If there's a disease on this one, males do not have another one, so the disease will be exhibited here. The disease will be masked here. But mom can still be a carrier. She can pass this chromosome off to her children. If she passes this chromosome off, her children won't have the disease. Okay? Males, if the male passes this one off, the child will have the disease. If the male passes this one off, the child will not have the disease there. It's what we call sex-linked. And where did that go? Right there. So X-linked traits mainly affect males because they don't have that extra X chromosome to cover it up. The Y-linked traits are only passed from father to son. So you have a look here. Heredity and dominance. And it's what we were talking about there with the big B's and the small B's. You have a look here. Here you have an individual there with brown eyes and blue eyes. Okay? And brown is the dominant. That's what the child will look like. If the mother passed on brown, big B, and the father passed on small b, this individual here will be big B, small b, and they'll have brown eyes because the big B, brown eyes, is dominant. Blue eyes, small b, is recessive. You need two of them. And you see there, small b was sent from mom, A small b was sent from dad, and now the child there has small b, small b. That is the code there for blue eyes. So dominant traits, there's lots of them. Widow's peak, okay, that's the, it occurs there on the forehead. The hairline comes down and dips down a little bit, kind of like, as my daughter would say, Count Dracula. All right. Nothing against vampires. I love them. I love all people. Actually, vampires aren't people. So, all right, whatever. Freckles, absolutely inherited. If mom and dad have freckles, child will have freckles as well. Dimples, okay, on the chin and the cheek, they're all inherited. So dominant disorders are uncommon because they are lethal and result in death before reproductive age. So anyone there who gets a dominant disease, okay, they're usually dead by the time, they hit puberty, because these are so destructive. Huntington's disease is caused by a delayed action gene associated there with the 4th chromosome. Most genetic disorders are inherited as simple recessive traits. Okay? Dominant diseases tend to kill the individual fairly early on. Recessive diseases, though, remember, in order for a recessive disease, okay, to be shown or demonstrated or visible in an individual, you need both of them. Okay, so recessive disease is kind of like, it works the same fashion as this. Like if the dominant gene is non-disease and the recessive gene is disease, it won't, in this case here, if the child gets both of them, it won't be evident. Okay, the child will not have the disease, but it could be a carrier. In this case here, if the disease is a recessive gene, and in this case here, if both parents donate that diseased recessive gene, then the child will have it. Okay, they will have the disease, what we call a recessive linked disease there. Albinism, okay, cystic fibrosis, Tay-Sachs disease. These heterozygotes are carriers who do not express the trait but can pass it on to their offspring. Okay, it's like what I was saying there, like male pattern baldness. Okay, the whole idea there that it is brought on by the mother. Okay, and generally speaking there, the reason why males go bald is because the disease or the imperfection there in the DNA came from the mother, and the father donated a Y chromosome, which there's nothing to mask that X with, so that's why the disease becomes evident in males and not females. Okay, this is where females there have the edge, evolutionarily speaking. Heredity dominance there, the dominant gene expresses itself overshadowing the recessive gene. Okay, hopefully there you guys have got that. The recessive gene is unexpressed if paired with a dominant gene, right? You remember there that this individual right there, they've got a gene for brown and a gene for blue. Brown is expressed because it's dominant. Blue is not expressed. It's in there. You can pass that blue gene on to your children, okay? But you can't see the blue in there because the brown is expressed. The blue is not, because the blue is recessive. Works the same way there when you're talking about diseases. The recessive gene is unexpressed if paired with a dominant gene. Codominant genes, though, oh yeah, there's all these different changes there. There's dominant, recessive, there's codominant genes. There's genes there that work together. This one here, there's also incomplete dominance. And this is an example here, sickle cell anemia, characterized there with low oxygen levels. It's quite debilitating to have sickle cell anemia. It's not, if you had the choice, you wouldn't want it. This is where the shape of the red blood cells are distorted, kind of like a sickle or like a crescent moon. And these RBCs, they get banged around and bent, and they only live about 3 to 4 months. Okay, but they have a certain shape for a reason. And when the RBCs are, when you have this disorder, sickle cell anemia, this is what the RBCs look like. And now they can't get down the capillaries very well. They're getting clogged and caught and all this kind of stuff there. They're very fragile, short-lived, and can clog up there the blood vessels. It's not a great thing to have, except if you were in, tropical regions, okay? Because having sickle cell anemia prevents you from getting malaria. People who get malaria, individuals there with sickle cell anemia do not suffer the consequences of malaria. Only people there that have the RBCs that are round, okay, the normal looking ones there. So you'll see here that big S, this is normal hemoglobin, normal RBC, okay, dominant. You have a look here. Here I have dominant, and there's that sickle cell recessive gene, okay? In this situation here, it's kind of like this one right here, where this person has brown eyes, but they've got a blue gene in there too, right? And you'll see that this one here, sickle cell trait. Both aberrant and normal hemoglobin are made, can suffer a sickle cell crisis under prolonged reduction. Okay, so these ones here are not that bad, okay, because they have that dominant there that's kind of protecting and overshadowing, okay, that recessive sickle cell gene. In this case here, though, there you have the recessive sickle cell and the recessive sickle cell, and that's all you have. So this person here is 100% sickle cell anemia. It only has, this individual only has this kind of RBCs, and they have sickle cell. So this kind of shows you the whole idea of dominance and recessive, okay? You've got, in this case here, this individual is double dominant, so they will only be able to pass on, okay, the same, all right? This person here, they, on the surface, they're okay, but they have that sickle cell gene. They could pass that off to their children, or they could pass that one off and then the sickle cell is done, right? Never to be seen again in this lineage, all right? But this individual here, if they choose to have kids, they will 100% pass either this one or that one, whatever it is, off to their children there, the recessive sickle cell gene. Last slide of the whole course. Are you ready to go? Sickle cell's trait there provides a survival advantage over people with normal hemoglobin in regions where malaria is endemic. This trait is known to cause specific fewer deaths due to malaria, especially when Plasmodium falciparum is the causative organism. Okay, so but they're just saying there that an individual there with sickle cell doesn't get malaria as bad as somebody there who has sickle cell. They have the double recessive. Someone who doesn't, they have the double dominant. And people there with sickle cell do not get malaria. That's what we've got going on there. And I'm going to stop sharing now. See if there's still, I can still see you very good. Well, that is it, people. You are done. You should give yourself a round of applause. Oh, stop it, Nicole. I'm just a teacher. I'm just a guy. And but thank you so much. You guys have been awesome. You really have been sticking it out in this crazy VR platform. And all I have to say is that I'm so proud of you guys. You guys there, just keep on going. You're doing well. The marks are doing great. You're all gonna make it, okay? And that's what we want. You just keep going on, and you guys have my email address there. Okay, and I'll type it in here. Ian.coates@georgiancollege.ca. There we go. You guys email me anytime from now until the next 50 years. All right? Because I'm 50 now. I don't plan on making it past 102. 102 is good if I can make it there to 102. So I'll be at George until then. Let me know if you guys are having any issues, but I think you guys are absolutely wonderful. Okay? Take care. Do not stop at a PSW. Get your PSW. Go on and become a nurse practitioner. Rule the world. And then when you're ruling the world, give me a call. All right? But take it easy, guys. Be well. I'll be in contact. I'll post this video there on announcements there. And test is next week. Assignments will be marked, hopefully there this weekend. Take care. You're almost done. But do not start celebrating yet. You got a week and a half. And then you got 3 weeks to kick off and relax and have some fun. But don't you dare take your foot off the gas. You've got 10 days until school is done. You just stay focused. You get your stuff done. And I'll see you at the finish line. Got it? Bye, guys. We'll see you later. Bye-bye.